everyone, and welcome to Day 3 of the 10th Annual Microbiology Virtual Event Series, brought to you by Labroots. Today's keynote presentation is titled Microbial Life in the Uranium-Contaminated Subsurface of the Oak Ridge Integrated Field Research Site. I'm Cassie Soltman of Labroots, and I'll be your moderator for this webinar. As a reminder to our audience, we encourage you to chat with your peers using the group chat window on your screen. You can also use the question submission window to submit questions for our speaker to answer live in the Q&A segment following the presentation. Now let's get started. I now present today's speaker, Dr. Stefan Green, Associate Professor, Infectious Diseases, Rush University. To view Stefan's complete biography, please visit the presenter window. Stefan, you may now begin your presentation.
Great. Thank you so much, Cassie. Really appreciate that. Good morning, everybody. Really happy to be here. Glad you're here as well. I'm happy to present today on work that I've been involved with it over the past probably 10, 12 years, or maybe even longer, about what I like to call the worst place in the world, but good for microbiology, the Oak Ridge Integrated Field Research Site, which, as explore in the next slides, is a really wild environmental location for conducting microbial research. Since I tend to talk a lot, I always like to do the acknowledgments right at the beginning. So this was Department of Energy funded research through the lab of Dr. Joel Koska, at that time at Florida State University, now at Georgia Tech. And then a number of other key people involved in the research are Dr. Pooja Jasrotia on the far left there, and Dr. Om Prakash, who is the main microbiologist doing cultivation work on the project, and Daniela Hubbard did a lot of the quantitative PCR. We also got funding from the Joint Genome Institute for doing bacterial genome sequencing, and some of those results you'll see as well. Okay, so just a quick outline of the talk. I just want to introduce you to this this really wild site in Oak Ridge, and then talk a little bit about mechanisms by which we can try to remediate sites like this. And at Oak Ridge, we'll see that actually the nitrogen cycle becomes extremely important due to inhibition of uranium remediation in the presence of nitrate. And then we'll explore a bit more the ecology, the microbial ecology of the site, both groundwater and sediment. And then I want to introduce you to a very interesting group of microorganisms, bacteria from this genus called Rhodanobacter. And then hopefully, if I haven't talked too long, we'll be able to talk a bit about denitrifying fungi in the subsurface as well, and then a little bit of summary. Okay, so let's talk first a little bit about why we have sites like this. These are all derived from uranium enrichment as part of nuclear weapons production, where you're looking to enrich for the fissile isotope of uranium, U-235, but that's a fairly low abundance isotope, so you end up generating a lot of uranium waste. And it's a global scale problem, but also there are a large number of sites in the United States, and the Oak Ridge site is just one of many. And we have this environmental remediation sciences program to support strategies to remediate these sites, hopefully using microorganisms. So just to give you a feeling, this is a site in Tennessee. It's quite a large site, and the waste that was generated during the uranium enrichment was dumped into these unlined disposal ponds called the S3 ponds. They're no longer there. They've been drained, and the ponds have been capped. So in the left here, you can see an aerial photograph. There's now a parking lot there. In fact, the first time I went to the site, there was a hot dog cookout right on the site, and I was like, my God, like, do you know what's underneath the pavement here? But basically, the site received just a massive quantity of nitric acid waste together with heavy metals, including uranium and other and also hydrocarbons. And because these sites use this and other sites have used unlined pits, basically, the liquid material starts to leach into the subsurface and is the source of a contaminant plume. So something on the scale of 1 trillion liters of nitric acid were dumped into these ponds. So we have a contaminant source at the site of the former ponds that's now in the subsurface, and there's groundwater that, of course, flows through the area, and then that migrates downstream to other tributaries and water supply for the local area. So there's quite a lot of interest in the remediation. Part of the issue and difficulty with these sites is that in this particular one, in addition to high uranium, so we're in the micromolar level of uranium waste, there's just absurd levels of nitrate. So in some cases, we have in the close to the source zone, on the scale of tens, hundreds, or even thousands of millimolar nitrate, which is a high, very, very high contamination levels. Now, the original liquid in the ponds, it was highly acidic. Again, nitric acid was basically used as a cleaning agent for some of the instrumentation, and so you have the ponds were extremely low pH. They lucked out a little bit with carbonate rocks underbedding the ponds that have ameliorated the pH. So we in the close to source zone, the pH is roughly in the area of 3 to 4. Still acidic, but could be a lot worse. And in addition to uranium, there are other heavy metals. So I like to say, at least from a human perspective, the subsurface here, so our groundwater and sediment are should be considered extreme environments. For microorganisms, what we consider as extreme is not always the case. So one of the things that we've been interested in is whether we can use microorganisms to catalyze reduction of uranium in a way that can serve a bioremediation strategy. So in the uranium, the oxidized uranium, uranium-6, is soluble in groundwater, but if bacteria are able to reduce it through as a terminal electron acceptor, then you can form an insoluble uranium mineral called uraninite, which will then precipitate out of groundwater and stop moving along the gradient. And so this is conceivably a nice idea, but there turned out to be a number of limitations to this. So first is that as far as we know, there are no what we call obligate uranium reducers, meaning there are no organisms that can only make their energetic living through using uranium as a terminal electron acceptor. And so many of the organisms have that are able to use uranium as an electron acceptor can also use many other compounds, and many of those other compounds are energetically more favorable than uranium as an electron acceptor. So in the presence of other compounds, then you don't actually reduce the uranium, and it stays soluble, oxidized and soluble. So it turns out that both oxygen and nitrate can serve as better alternative electron acceptors relative to uranium as a terminal electron acceptor. In addition, even if we do precipitate uranium as uraninite, it's not a stable mineral and is highly labile and can be reoxidized and resolubilized, and then it can continue to migrate in the groundwater. But if we do want to try to remediate uranium using a microbially mediated mechanism, we really need to deplete both oxygen and nitrate from the groundwater. And given that we're in the tens, hundreds, thousands of millimolar nitrate, it's a challenge. So one of the things is we became quite interested in microbial mediated nitrate removal, and not just at Oak Ridge, but in other locations, nitrate is a very serious groundwater contaminant. But luckily there are microorganisms that can catalyze the conversion of nitrate to nitrogen gas, and then that can basically replace a harmful compound with a, or potentially a harmful compound with a inert gas. But as you'll see during the talk, there are other intermediates in the what we call the denitrification pathway, so conversion of nitrate to nitrogen gas, that can be produced, including nitrous oxide, which is a serious greenhouse gas. So there are other considerations as well. I just want to talk a little bit about denitrification, since a lot of the talk really will focus on this process. We start, of course, with nitrate, and there are a series of enzymatic reductions of the nitrogen atom from +5 oxidation state to 0 in this pathway that we call the dissimilatory denitrification pathway, meaning conversion of nitrate to nitrogen for energetics. But there's also a pathway for reducing nitrate all the way down to the level of ammonium. But that's what we would call an assimilatory pathway, meaning that you're reducing the nitrogen to the level of the carbon of the molecules in cells. And what we're really focusing is on energetics for dissimilatory denitrification. So overall, if we just kind of look at a very basic chemistry, this is not the only coupled reaction that you can have for denitrification, but basically we can take denitrifying microorganisms can take organic carbon, use that as their carbon and electron source, and oxidize the organic carbon to carbon dioxide. And at the same time, we couple that to the terminal reduction of nitrate, ultimately to nitrogen gas. And there are a number of different microorganisms from different lineages that can catalyze this reaction. We also can find at least parts of the pathway in all domains of life. So there are eukaryotic organisms. We'll talk a bit about fungi and their capacity for denitrification. Of course, these organisms that are able to denitrify tend to be facultative anaerobic organisms, so they can tolerate both aerobic and anaerobic conditions. And one of the things that's come out of extensive work into the ecology and phylogeny of denitrifying organisms is that that the genes involved in denitrification can be laterally transferred or horizontally transferred between lineages. So the phylogeny of genes involved in denitrification may not match that of the host housekeeping genes, such as the ribosomal RNA gene. And I should just also mention that there are basically two basic forms of one of the key enzymes in this pathway called the nitrite reductase genes, so that there can be both cytochrome type and copper containing nitrite reductases. We're going to be focusing on the copper containing nitrite reductases in the organisms that we'll be looking at. Okay, so back to Oak Ridge, and here's just a little diagram of the site. If you'll see here, there's a kind of a little square. That's our former S3 ponds, and there's two basic pathways for groundwater to migrate across the site. One is what's called the shale pathway, and as you go down the shale pathway, you see that the pH actually increases, so you're back to more circumneutral pH. And then there's also this carbonate pathway that dives deeper into the subsurface. And one of the things that we were interested in is whether there's active denitrification in the site. Are the native microorganisms catalyzing this reduction of nitrate to nitrogen gas or any of the intermediates? So one of the things you can do is just in the headspace of the wells. So they're wells that have been emplaced in multiple locations across the site. You can measure headspace gases in those wells, and one of the obviously byproducts of denitrification could be nitrous oxide. And so what you'll see is relatively high levels of nitrous oxide in the wells. That's these yellow bars here. You see quite high levels of nitrous oxide near the source zone, near the S3 pond, former S3 pond, and then as you go down this shale pathway, as the pH rises, you start to see less and less nitrous oxide in the headspace. And this is pretty well known that at low pH, you tend to favor the production of nitrous oxide rather than complete denitrification all the way to nitrogen gas. Okay, so we had a fairly simple experimental approach in the main studies that we were doing at the site. We were doing both groundwater and sediment sampling from the site, and we basically had two different pipelines of analyses. One were on the molecular biology, cultivation-independent molecular analyses, including quantitative PCR, high throughput next generation sequencing of amplicons targeting 16S ribosomal RNA genes, and also some metagenome sequencing. But at the same time, with Dr. Prakash, we were also running a cultivation pipeline looking to isolate relevant denitrifying organisms from the site. And then the idea was to integrate those molecular and isolate data, in part by also from isolates performing whole genome sequencing. We were particularly interested in trying to identify the genes in the denitrification pathway as well. And of course, there's also geochemistry associated with these analyses. Okay, so I want to start a little bit with the isolation work, cultivation of denitrifying organisms. Dr. Prakash was able to isolate quite a number of denitrifying bacteria from the site, and we've actually used them both for genome sequencing, and also have deposited some of these in the German type culture collection. And I just want to point out a few features of these organisms. So we have both what we would call proteobacteria, gram-negative denitrifiers, but also some gram-positive denitrifiers from both the former Firmicutes and actinobacterial phyla. Sorry, they've been renamed, and I'm still not used to using the new names. But one of the things that you can see is we looked, at least under experimental conditions in the laboratory, we looked for the terminal nitrogen species that we were generating through the denitrification pathway. So we have a number of alpha proteobacteria, for example, that the terminal denitrification is nitrogen gas. Whereas an organism which you'll hear a lot about, this Rhodanobacter, is producing a mixture of both nitrogen and nitrous oxide, whereas the gram-positive bacteria are producing nitrous oxide as the terminal product and don't produce nitrogen. Okay, so looking at the ecology of the subsurface, we had a large number of groundwater wells to analyze. So both wells that were close to the source zone, low pH, high nitrate, high uranium levels, and then across a gradient across the site as you go along, for example, down the shale pathway, the pH starts to increase and the concentrations of nitrate and uranium decrease. So what we're looking at here is just a multi-dimensional scaling plot, ordination plot of 16S ribosomal RNA gene amplicon sequence data for a number of these wells at a single sampling event. And just for those of you who are not that familiar with these, each symbol or each dot represents a single sample and the entire microbial community in that sample, and the distance between any two of those points is a measure of how dissimilar the two samples are in terms of the total microbial community. So data points that are symbols that are quite close together represent two samples that have very similar microbial communities. So that's our sum total of samples. If we now color code these by pH, we see that there's clustering of a number of the wells that have pH below 4 relative to the rest of the wells with pH greater than 4. And again, this makes sense. These are the wells that are closest to the former S3 ponds, most highly impacted by the plume. We would expect more similar microbial communities due to the kind of extreme nature of the environment. But if we overlay that also with the relative abundance of bacteria from the genus Rhodanobacter, we also see that many of those highly impacted wells also contain relatively high relative abundance of bacteria from the Rhodanobacter. So this was kind of our first hint that these organisms were relevant in the subsurface, and at the same time we also had isolates from the same genus from the same wells. So we also wanted to look to see whether these organisms were active under the in situ conditions and not just dead DNA that's migrating down with the gradient. So we also extracted RNA from the source zone wells. So these are 5 different wells in the near source zone. And then we use quantitative PCR with primer probe assays, looking for total microbial community abundance. And then we developed a Rhodanobacter specific assay. And what we found is that in many of these wells, the vast majority of the RNA, the ribosome RNA that we were detecting was from Rhodanobacter. So in some wells, for example, 99% of all the RNA that we detected that we quantified was Rhodanobacter. So not only are they highly abundant in the source zone, they're also active under the in situ environmental conditions of low pH, high nitrate, high uranium. So when we looked at quantitative assays across the site, not just in the source zone, and sorry for the a little bit messed up y-axis legend here, but basically on the y-axis we have it's a log scale of the absolute abundance of ribosomal RNA genes per liter of groundwater. And then across the x-axis is a pH going from a pH of about 3 to about 7. And in black symbols, we have the measured abundance of total bacteria, so a pan-bacterial domain assay. And one of the interesting things is we really didn't see much of an effect of pH. There's almost no slope, in the absolute abundance of total bacterial load at per liter of groundwater. But when we look at our Rhodanobacter specific assays, here we see a strong negative correlation with increasing pH. So the absolute abundance, not just the relative abundance of Rhodanobacter, is highest in the low pH source zone, and then they become less and less abundant as the pH increases across the site. So we also looked at not just at groundwater, but also at the sediment. This is again microbial community analysis using 16S ribosomal RNA gene amplicon sequencing, and we have some samples on the left side of our PCA here that represent samples that are non-impacted, so basically not close to the source zone, quite low nitrate levels, and they have a distinct microbial community that has a quite high microbial diversity, as you might expect in a natural groundwater. But on the right side of the plot, we have what we call the plume-impacted sediment samples, and here we have a number of samples from extremely high nitrate, either 1 to 10 millimolar in orange and over 10 millimolar in red, together with a slightly less nitrate-impacted sediment sample. And in both of these, particularly in the slightly lower nitrate sample, we see incredibly high levels of Rhodanobacter also in sediment samples, ranging from up to 3/4 of the total 16S amplicon sequencing data that we have. So the Rhodanobacter are found not just in the groundwater, but also in the sediment, higher in the impacted source zone, and much lower in the both in absolute and relative abundance in the more ameliorated locations at the site. And so, because this organism seemed to be so important at the site, we started to get quite interested. We also, as I mentioned, had a number of isolates that Dr. Prakash had generated. And so we dug in a little bit to see what we could find out about this group of organisms. So they belong to the gamma proteobacteria and a family called Xanthomonidacea. Currently, there are about 20 described species. When we started the work, there were probably 8 or 9. And so a lot less was known about their ecological niche. And as I mentioned, we had isolated a number of different isolates from the site, some of which were denitrifying to nitrous oxide and to nitrogen gas. But we found that no matter what pH that we grew them at, they were quite slow growers, and never, even at optimal conditions, never really were what you would call a fast growing E. coli style proteobacterium. So we did do a lot of characterization of the isolates and described a new species called Rhodanobacter denitrificans. So one of the great things about denitrifiers is that usually you stick it in as the species name, and it helps you a lot in trying to know what what the organism actually does. And just a few notes about the kind of other physiological characteristics that we were able to characterize. So it is fairly flexible in terms of carbon substrate that it can use for growth. And it has also quite a strange cell dimensions. I'll show you a micrograph in just a moment, but very long and thin cells, but motile. And here's a micrograph that we generated from one of the cultures. So this was even cells growing on a rich medium don't really produce kind of very plump bacteria that you might come to think of as from gamma proteobacteria. We at the time were able to get the Joint Genome Institute to very thankfully sequence the genome of our type strain. So this 2-APBS1 isolate, and in analyzing the genome, it has fairly standard microbial characteristics. So it can do glycolysis and gluconeogenesis, has a TCA cycle. And importantly for us, critically, it has genes involved in denitrification, as we might expect. But one of the interesting finds is that it actually had 2 genes involved in nitrite reductase. So we'll go over that a bit more in a second. It's the second step in the denitrification pathway, where we're converting nitrite to nitric oxide. So it's got actually two different genes located in different locations in the genome. And then some indications of how it's able to tolerate the quite low pH levels in the source zone. It's got a decarboxylate, arginine or ornithine decarboxylase, and also antiporters for sodium and protons. So when we look at the genome characteristics, so this was from a group we were working with. Chris Hemme wrote a very nice manuscript describing metagenomics at the source zone, and we were able to utilize our genome of the 2-APBS1 strain. So it's probably a little hard for you to see, but there are 2 features that I just want to point out. Is genes involved in denitrification are highlighted in green, and then genes that are likely lateral gene transfers are highlighted in these kind of brown colors around the main chromosome of the organism. And then I'm not sure if you can see this, but here's one of the nitrite reductase genes is located in association with many of other genes in the denitrification pathway, but a second nitrite reductase is found up here, and there are a few other genes involved in denitrification that are located elsewhere in the genome. And then you'll also notice there are kind of hotspots for lateral gene transfer, and these seem to, at least some of them, to be affiliated with metal metal resistance genes. Clearly the organism has genetic capabilities that are commensurate with the situation in the groundwater, with both presence of heavy metals and also high nitrate. So we had isolated a number of different organisms, and as I mentioned, some of them were denitrifiers and some were not. And we also ordered a number of other isolates from the American type culture collection that had been previously deposited. And what we found in our hands is that, again, a few of them were denitrifiers. So, for example, our type strain, Rhodonobacter denitrificans 2-APBS1 is a denitrifying organism. And then, for example, there was a Rhodonobacter thioxidans that we'd gotten from another from the ATCC that was also a denitrifier. But other members of the genus were not denitrifiers in our hands. And then also one of the isolates from the subsurface was also not capable of denitrification under the conditions that we were growing them. So when we sequenced the genomes, the story became quite clear that really there was no mystery here. They're actually all denitrifiers with full denitrification pathways, with the exception of the first gene in the pathway, the nitrate reductase, so conversion of nitrate to nitrite. So the organisms that didn't denitrify in our hands were ones that were missing that first nitrate reductase. And in an environment like the Oak Ridge subsurface, there's so much nitrite in any case from organisms that do have the nitrate reductase that it's not really a limitation. So we think that denitrification capability is a general feature of the genus. When we look at the pathways, the gene order or synteny of the genes involved in denitrification. So, for example, this is a gene involved in nitrous oxide reductase, so the final step converting nitrous oxide to nitrogen gas. There are a number of genes involved in this. The gene order between our organism, the type strain 2-APBS1, is that first row, and the most closely related organisms basically have the same gene order. So we feel that indicates more of a homegrown, not laterally transferred gene. But then when we look at the nitrite reductase, so this is just one of the two genes. I don't want to belabor it too much. When we look at this, our organism, there's just a single gene that provides the functionality for nitrite reductase, and there's just no gene synteny with other organisms that have similar genes. So this is much more probable of a lateral gene transfer from other organisms. One of the things that this did create a problem for us is when we were initially analyzing the site, we were never detecting Rhodonobacter genes in surveys of genes involved in the denitrification pathway. And one of the things that we realized once we started to have genome sequence data is that the primers we were using, very general nitrite reductase primers, for example, basically are only targeting these organisms that are in this orange clade here, and those represent kind of more traditionally Pseudomonas style denitrifiers. And the primers have very few mismatches. The mismatches are indicated in red. A perfect match between primer and the genome sequence is indicated in green. And then over here we have the inferred amino acid sequence that the primer is targeting. And we'll see that the primers for that group of orange organisms, amplifies those organisms very well. But our Rhodonobacter are way at the top, up in this green group of clade of organisms on our phylogenetic tree, and those have a tremendous number of mismatches between the primer and the genome. But there is a conserved histidine residue that's actually involved in the copper binding of the nitrite reductase. So at least that amino acid is conserved, but that's not enough really for a primer. So we really had to develop new primers based on the genomic sequences. So we now kind of moved on to more general physiology of multiple isolates from the Oak Ridge site. We had isolated them from a number of different wells that had different pHs. And we looked to see whether they were able to grow on nitrate, and at least in the table that I'm showing here, all of them were able to grow, but only some of them were producing nitrous oxide. So that even though they all have nitrous oxide reductase genes, they're not all working at the same rate under the in situ or experimental conditions. So there are still important physiological differences between these organisms, isolates from the same genus. When we looked at their tolerance for heavy metals, we found that for the most part, they were in excess of what is found at the site. So for example, and just be careful here, the minimum inhibitory concentrations, or MICs, are shown in millimole per liter, whereas the concentrations in the Oak Ridge site are in the micromolar range. But for example, for uranium, the concentrations, 10 to 66 and as high as 155. But the MIC for uranium for a couple of our isolates, for example, we're on the scale of 500 micromolar, so well above what we find in situ. And then, similarly in terms of nitrate, again, we can the wells that we had tested, up to on the scale of 200 millimolar, but at least our type strain and one closely related isolate, could handle 400 millimolar nitrate and 100 millimolar nitrite. So, extremely high tolerance for these compounds. One other very interesting thing is they seem to grow very differently given the experimental conditions. So a couple things to note here. These are just growth curves for the type strain. And just notice the very long scale here. So this is time in hours on the x-axis, and then OD 600 on the y-axis. And they're very, very slow growers. But one of the things is if we grow them in a rich medium, so this R2A medium, one of the things that we found is if you start the pH around 4, they will increase the pH during growth. But we found that when we used a synthetic groundwater medium that was much simpler, lower organic content, but supplemented with dextrose, that we see a decrease in pH from about 4 to below 3 during growth under those conditions. So we think that obviously growth conditions are important, but that also indicates that the pH tolerance is probably broader than we think for these organisms. Okay, in the last few minutes here, I want to turn our focus from Rhodonobacter to some work done by Dr. Pooja Jasrotia at the site, and when we looked at fungal presence of fungal denitrifiers at the site. We were interested also because there is some evidence for fungal denitrification, but that they're not capable of nitrous oxide reduction as far as that is indicated in the literature. But that also fungi can be metal resistant and can be found in an acidic environment. So, Oak Ridge again would seem to be a plausible environment for them. So this is another abundance plot on the y-axis is the log ribosome RNA gene abundance per liter of groundwater, pH on the x-axis. The black symbols are bacteria, and the open circles are fungi. So there is a slight trend towards increased relative abundance of fungi with elevated pH. They're rarely as abundant as bacteria. So these are not as highly abundant in the subsurface, but still present. But we do see also this patterning. So that this is a canonical correspondence analysis, but basically the take-home message here is the wells or the samples indicated in red are more similar to each other relative to those in green. So again, the red represent the acidic near source zone subsurface with pH below 4.5. So Dr. Jazrotia was able to isolate a number of fungal isolates. And when we grew those in pure culture, initially in capped microcosms, initially with oxygen-containing headspace, we see that again slow growth, and then as the oxygen is consumed, then you can start to see the production of nitrous oxide by many different fungal lineages in the subsurface. But as I mentioned, as far as we know, fungi are not able to do the terminal step with the nitrous oxide reductase. So then this is again a source of a potent greenhouse gas through denitrification by these organisms. Okay, so thanks for bearing with me. Just to kind of summarize everything we've gone over. One of the things that we see that pH is a major driver for microbial community composition at the site, and also is co-associated with other contaminants, including nitrate and uranium and other heavy metals. And it seems like a combination of this pH and elevated nitrate are highly selective for bacteria from the genus Rhodanobacter, which we can see that are capable of complete denitrification. And I'd also like to just highlight that the value of kind of iterative both cultivation and cultivation-independent molecular approaches, the two go very well hand in hand. So we never would have realized that the denitrification capabilities of all the was a general feature of Rhodanobacter without the genome sequencing that indicated the absence of a nitrate reductase from some of the strains. And then just a note that primer bias continues to be an issue where you can miss critical organisms just because your primers aren't targeting a broad enough range of organisms. This is not just true for denitrifiers. And then, Rhodanobacter is an interesting organism, has a lot of laterally transferred genes. We're not really sure what the role of two different nitrite reductase genes is. So there's future work and also the decision making on whether to produce nitrous oxide or to complete denitrification to nitrogen gas. And then finally, fungi, though less tolerant, it seems, of the subsurface contamination, are still capable of denitrification in the subsurface environment. And then overall, just a reminder that we don't want these partial denitrification byproducts. We really would like to be able to push organisms to complete denitrification to nitrogen. And with that, I greatly appreciate your attention and happy to answer any questions.
Thank you, Stefan, for your informative presentation. Let's go ahead and dive into the live Q&A. Remember, you can submit your questions below for our speaker to answer live. We have a couple of questions that have come in so far, so I'll just go ahead and jump in. Our first question asks, where else are Rhodanobacter species found beyond Oak Ridge?
Yeah, so this is a really nice question. So there are, when you look at the kind of 20 different species of the genus, there seem to be two types of Rhodanobacter that are recovered. Some seem to be fairly standard soil microorganisms that are found in rhizosphere plant-associated environments, but others are clearly found in acidic nitrate rich environments. So there's one study showing it denitrification at pH below 4 in a bioreactor. And I think it's clearly one of the capabilities. Under the right conditions, they can grow to very high relative abundance, but they don't seem to be particularly abundant in other environments. So they do, when they find their niche, they can really exploit it, but they don't have so many niches.
Wonderful, thank you. All right, our next question here asks, how can new molecular tools help study contaminated environments?
Yeah, one of the so it's a great question. So one of the things to remember is that Oak Ridge is a bit of an unusual environment. Not every uranium-contaminated environment has so much nitrate, and so I think you need to be able to tailor your approach to characterizing these environments um, accordingly. So the molecular tools can be really valuable, particularly, for example, shotgun metagenomics, which is a method that's basically agnostic to the organism. So you don't need to design primers. It will basically just sequence everything that's in the sample, and then that can give you genetic information that you can then use to design quantitative PCR assays, to design culture conditions that can help you enrich the most abundant organisms in those samples. So I feel that the molecular tools give you a guide to ways to interrogate the site that you're working at, and since each site is different, it's really important to use those cultivation independent tools initially, and then open up opportunities for both cultivation and higher throughput analyses of multiple samples across any given site.
Wonderful, thank you. All right, moving on. We have another question here. Can Rhodanobacter or other dendrifying bacteria be used for remediation of contaminated environments?
So it's a really good question. I haven't seen anybody using it. One of the downsides with Rhodanobacter is that it does seem to be fairly slow growing. So it's not necessarily a super ideal organism. And again, it has to have very specific niche for it to really thrive. So it's probably not a good global organism for denitrification remediation. But certainly, you can use denitrifiers as a way to remediate contaminated subsurface environments. One of the things that was kind of interesting at Oak Ridge, kind of concurrent when I was working there, is that they were trying to use bioremediation to remove the uranium. And so they basically were taking a pump and treat approach where they would pump the groundwater out from an area just before the area that they were trying to remediate, and then treat to remove the nitrate from the groundwater, and then re-inject the treated water back into the subsurface, and then you would feed it carbon and then let the uranium reduction go forward. So you can use, for example, denitrifying organisms in the bioreactor to remove nitrate from samples. So under the right conditions, I think you probably could use Rhodanobacter, but it's not a global organism that would work well everywhere.
Great. All right. We have another question here. It asks, has Rhodanobacter been identified as an etiologic agent in disease?
I have not seen any papers showing it as a pathogen or even a putative pathogen. It doesn't seem likely that it would find a good niche inside you. So I don't think there's any pathogen concern here. I will say one kind of on a tangent here. They do seem to be amenable to genetic modification, and I'm hopeful that these will become laboratory rats of the way that E. coli are, because it is quite an interesting organism. But I don't think there are any pathogenicity concerns with it.
Good. Okay. All right. Our next question here asks, have you thought about transferring nitrate reductase genes to dendrifying fungi?
Okay, so this is actually a very interesting question. I myself haven't, and I think the question would be whether it's worth it if there are organisms that can already do the terminal denitrification step. But I will tell you a little bit of a side story there. I think there's another speaker today, Dr. Dror Mintz, who actually was my PhD advisor, who's working in agriculture fields, agricultural systems, where nitrous oxide can also be production can also be of concern. And he's been working to isolate organisms that are basically only capable of nitrous oxide reduction to nitrogen, so they don't do the other stages in the denitrification pathway. And the idea would be to amend them to plant roots, for example, as a way to reduce nitrous oxide production in agricultural soils. So I think there is a lot of interest in genetically modifying organisms to suit your role. And I think it could conceivably be possible, but I think it would take some work to get a bacterial gene working well in a eukaryotic organism. So I'm not sure if that's been tried, but a very interesting idea.
Great, thank you. All right, our next question here asks, what are the next steps in your research on microbial life in contaminated subsurface environments, and are there any new areas of study or emerging technologies that you are particularly excited about?
So my latter career has been moving away, unfortunately, from contaminated subsurface environments. And so now I'm really much more involved in host-associated microorganisms in human health. And so one area of study that I'm working on right now is looking at the effects of space flight on gut microbiome in mammals, so mice and also humans. The data are not as mature as the work on Rhodonobacter, but hopefully next year, if I'm invited back to give another talk, I can give you some of the results from those studies.
Excellent, thank you so much. All right, next question here. How do oxygen conditions affect the physiology of subsurface organisms?
Yeah, so this is actually really, really interesting and also very important. So the microorganisms are going to alter their gene expression profiles to match the environmental conditions. So the presence of oxygen as oxygen is a highly favorable terminal electron acceptor for many of the organisms in the subsurface. Is almost always preferentially used while present. Particularly in sites where you have active remediation, usually you're also dumping electron donor into the subsurface through the either as some sort of organic carbon, could be acetate, something like that, where you dump that into the subsurface, and then organisms will consume the oxygen while they can, and then once the oxygen has been consumed, will then switch metabolism to use an alternate electron acceptor, such as nitrate. So you have this critical shift in a respiratory activity based on the presence of oxygen. It can, but even low levels of oxygen may still allow for the basically turning on of denitrification genes. And as you saw in for example, in the fungi, most of the denitrification activity is occurring once the oxygen levels have been drawn down. So many of the denitrifiers are what we would call facultative, meaning that they can also use oxygen when it's present. And so you basically got to get rid of the oxygen, then they'll turn to the nitrate to reduce the nitrate. It also can affect just in general conditions of stability of some of the labile uranium minerals as well. So there's lots of stuff that will change with the presence or absence of oxygen in the subsurface.
Wonderful, thank you. All right, our next question here says, do you foresee the insights gained from the Oak Ridge site being applicable to other contaminated environments, particularly those involving heavy metals and mixed waste?
Yeah, I will speak for myself. I think the statement I'm going to say is maybe a little bit controversial, so I don't want to give the feeling that this is absolutely true. But my feeling is one of the things that the study at Oak Ridge has shown us, at least in this particular site, is that it's extremely difficult to use active bioremediation to address the uranium contamination issue in the Oak Ridge site. And part of there are a number of factors that are really contribute to this. So one is just the tremendous amount of nitrate that's in the groundwater. We've already talked at length about it, but to even like temporarily remove nitrate from a small area of the subsurface is extremely like dollar intensive. It's extremely technically challenging and expensive. So it's not really a viable remediation strategy on any large scale or long-term scale. And one of the things that we actually see at the site, and people don't like to hear this, but that the old saying, dilution is the solution to pollution, actually might apply at Oak Ridge, because by the time the amelioration of the pH as you go down, for example, the shale pathway, the pH comes up, nitrous oxide is no longer being produced. The uranium levels are basically below, levels of concern. So that the site itself is actually able to kind of remediate itself. It's just that at the source zone is just such an alarming concentration of the contaminants that you do want to feel like you're actively remediating. So some of the lessons are actually, kind of negative lessons that in highly complex subsurface, like Oak Ridge, it can be very challenging, because not only do even if you're adding organic carbon, you have to be able to deliver that organic carbon to everywhere that the uranium is, and there's very complex geology in the site. So I think what it does suggest is that you really have to know your site, and you need to pay attention to the geochemical conditions at the site and the groundwater hydrology. All of those are essential pieces of any bioremediation strategy.
Excellent, thank you. All right, we have a little time left, so I'll just do two more questions here. This one says, what specific physiological mechanisms enable Rhodonobacter species to thrive in such highly contaminated environments, and are there any potential bioremediation applications being explored with these bacteria?
Yeah, so there are a number of features of the Rhodonobacter that seem to allow them to tolerate the subsurface. Part of it is they have a large number of genes involved in metal resistance. A lot of them seem to be laterally transferred. They do have full denitrification capabilities, or at least, not all of them have nitrate reductase, but the rest of the denitrification pathways. So certainly they seem to excel in acidic and high nitrate conditions, so that the denitrification pathway is essential for that. We haven't fully explored their pH tolerance. I would love to be able to do some RNA-seq studies to look at the differential transcription profiles under different pH. There's some indication that they can actually drive the pH down due to their own metabolism. That could give them a competitive advantage. So even though that they're slow-growing organisms, if they make the environment so uncomfortable for other organisms, then they can do well. And it seems to be just that that right combination of acidic and nitrate-rich environments that allow them to grow. So the genome is certainly consistent with that ability to handle those environmental conditions. But why they're able to grow so high, we don't have a good explanation. And for the second part of the question, I haven't seen so many bioremediation strategies using Rhodonobacter. And again, it seems to be in part because they have such a unique niche that it's not necessarily applicable to other groundwater environments that have high nitrate. You wouldn't expect, for example, nitrate contaminated groundwater to necessarily also to have a pH of 3.5. So that's the kind of the rare piece of here. But certainly they could be potential bioremediation agents if those the acidic conditions are also available. And so together the Rhodonobacter could do a good job.
Great. And before we go, the people need to know, did you try one of the parking lot hot dogs?
I did not try one of the hot dogs. I was actually horrified. That was my first time to the site. Really, like a few feet away were the sampling wells. I was just like, wow. I just didn't feel comfortable eating. But I guess you get used to it, right? So
Yeah,
it's just a parking lot now.
definitely. Well, Stephen, did you have any final comments to share with our audience today before we go?
No, I just really appreciate all of your time, and I'm happy to, if anybody wants to email me, I was happy to talk about Rhodonobacter, and just, kind of a shout out to all the other colleagues involved in this research. This was not a one-person project by any means, and then also thanks to the DOE for funding it.
Wonderful. Thank you, Stefan, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. Questions that we did not have time for and those submitted during the on-demand period will be addressed by our speaker via the contact information provided at the time of registration. This webcast can be viewed on demand for 1 year until September 5th, 2025. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Feel free to continue exploring this incredible virtual space, including the exhibit hall where you can check out our exhibitor booths and speak with their representatives. And don't miss out on earning points by chatting in the social space, viewing content, finding the hidden icon in each space, and more. The top 3 point leaders will receive a $25 gift card to the Labroots store. Thanks again to our audience and to you, Stefan. Goodbye, everybody.